My name is Åshild Ergon. I work at the Norwegian University of Life Sciences, and I am coordinating this project, Blue BioBoost. It is a sustainable blue economy partnership project, and we are partners from these 4 countries, both the research organizations and the seaweed industries. And we work with these 2 species, sea lettuce and sugar kelp. And I will just give you a brief overview of what we do in the project. One thing is that we try to find optimal light qualities to make the gametophytes grow faster. Then we have experiment running in 3 countries that I will get back to and tell you a little bit more about. And when it comes to ulva, we are working with so-called giant ulvas. So some individuals are very big, and we try to find out if they are big because they are polyploid, and if this is a heritable phenotype, and if it can be used in practice. And then we have developed some protocols for ploidy determination in both species. And finally, we have a work package for where we collaborate with stakeholders, and the goal is to make some best practice strategies for future European macroalgal breeding. So it's very nice for us to take part in this workshop and also collaborate with the Global Seaweed Coalition on this. Okay, so this is one of the questions that Melanie sent to us. How can seaweed strain selection and breeding contribute to climate resilient aquaculture and restoration while safeguarding seaweed genetic diversity? And you have already heard some talks on this already. So there are some technologies that may be solving the issue and preventing spread and genetic pollution of wild populations, like maybe we will hear later about sporeless kelp. Also sterile triploids could be maybe a solution. Another way is to breed, do plant breeding based on local material that we also just heard about from Sophia. But plant breeding can be quite expensive, so if we are going to have many local breeding populations, they need to be simple and cheap. And one simple and cheap way of breeding is mixed hybridization and mass selection. This is the kind of breeding that farmers have used many years ago when they gradually developed land races of agricultural crops. And we tried this strategy in the former project called Breed for Kelp to Feed, where we used the same kind of breeding procedure that farmers use. So farmers collect wild sporophytes and collect spores from them to generate plants for cultivation. And then we try to select the biggest ones and made them sporulate, and then mixed all the spores from all the different genotypes in one flask, so that all individuals could freely hybridize with all the others. And then we sewed it onto a rope again. So we performed only one cycle of selection, but it did not succeed, and we did not see any progress, actually slightly the opposite. And what we could see in this experiment was that there was a lot of selfings, so a lot of self-hybridization going on in this flask. And in our case, they had inbreeding depression, resulting in a worse crop the next year. Also, there was a lot of unintended selection going on. So we didn't only have the selection that we targeted here in blue, but also there were probably a lot of other things going on in this process, indicated with these different white arrows, where some selection that we didn't control occurred. So this was not successful when we tried. Of course, it was only one experiment and one generation. So in Blue BioBoost, we have another strategy. So here we make gametophyte cultures from single spores, so that all cells in the culture are identical. Then we make crosses between them, and then all the offspring sporophytes will be identical. And we aim to identify crosses that have a good performance and stable performance across years, and then we would in the future like to cultivate a mixture of such good crosses, which could be put together in a way that the general allele frequency spectrum closely resembles the wild population. So just a few more details. So in this kind of strategy, we would be utilizing both general and specific combining ability. And here you see what we have done. So we have collected wild sporophytes from each of these 3 countries, in Sweden, Ireland and Norway. So we have 1 location in each country with 3 sub-locations, and we have isolated single spore monoclonal gametophyte cultures from these sporophytes, and we put up a selection, now a crossing scheme, and now we are testing the different crosses in the sea. So here you see 2 stretches of rope with the offspring of 2 crosses. And then we will repeat this the next year to get a measure of how stable they are across different years. So the goal then is to create local mixtures of high-yielding crosses for cultivation with similar allele frequency profiles as the natural population. And this kind of strategy would be very low cost, so that it could be implemented by individual seaweed farmers. It would be low risk, as it is based on local populations and would have similar allele frequencies to the wild populations. But the success depends on a certain amount of variation in the performance of the different crosses, and that there is limited genotype by year interactions, because if we have crosses performing very well one year but not the next year, then it's not worthwhile. And these 2 things is what we are testing in Blue BioBoost. Yeah, we were also asked the question number 2, what regulatory and governance approaches are suitable? And I don't have much to add there, and we have already heard something about the possibility of modeling using population genetics. And I think we could learn a lot from modeling and simulations of introgressions, taking all these different factors into account, and also from monitoring spread from cultivated kelp farms. Okay, this is a list of people involved in the project that I want to acknowledge. Okay, thank you.